So thank you, Meek, for your work, for introducing me in such an excellent way. Thank you so much for your kind words. So we are going to start with CAR T cell therapy. I am welcoming you all to this webinar. So this CAR T cell therapy is actually it is a revolution in cancer treatment, and not only in cancer treatment. Now it is used in various types of autoimmune disorders, then diabetes, type 2 diabetes, allergy, asthma. For all the purposes now, this CAR T cell therapy is used. So this webinar, we will focus on what do you mean by this CAR T cell therapy? What does CAR T means? What is its application? What is its history? Which are the FDA approved drugs that are available? All we can discuss in this webinar. So please feel free to ask the questions, ask the doubts. Whatever possible, I will answer. So please do make it useful. Let's start. So before going or moving to CAR T cell therapy, I need to say something about immunotherapy. Can anyone just say what do you mean by this immunotherapy? Can you unmute? Because we can make this a discussion also. Then it will be, if you also involved, then it will be not boring for you. So can anyone say what is immunotherapy? Can anyone unmute and say what is immunotherapy?
It is related to immunostimulants and immunosuppressants.
Yeah, immunostimulants and immunosuppressants. And immunotherapy is the use of immunostimulants and immunosuppressants. So, what we can say is immunity. What do you mean by immunity? Anyway, you all know. So, we are using the what is the main cells that are involved in immunity? T cells and B cells. No. So, here in immunotherapy, we are using the T cells. We are activating the T cells and make it useful for. So there are different therapies available for cancer, and we all know that it is very difficult for treating the advanced stages due to so much complexity. There comes the role of this immunotherapy. Immunotherapy will boost one's own immune system through various mechanisms. And this immunotherapy, which mainly involves T cell therapy, is again divided into three. Have you heard of TIF? That is tumor infiltrating T cells. Then genetic engineering T cells. And the third thing is what we are going to discuss today, nothing but CAR T cell therapy. So in tumor infiltration therapy, what we are doing is we are just collecting the tumor from metastatic tumors. We are collecting the T cells from metastatic tumors. And we make some modifications and reinfuse to the patient itself. Coming to the complications or limitation is that the culturing or the separation itself is a tedious process. Itself is very tedious. So next immunotherapy is what is known as engineering, T cell engineering therapy. T cell engineering therapy, what we are using is modified T cells we are using. And here the mechanism of action is always, you may have heard of major histocompatibility complex. So MHC mediated mechanism. Next is CAR T cell therapy. So what is CAR T cell therapy and what is the advantage of this CAR T cell therapy? Here comes the advantage of CAR T cell therapy, that is, in case of certain tumors and majority of solid tumors, the MHC will be down regulated or deletion of MHC will be there. So CAR T cell therapy's mechanism of action is it boosts the immune cells through through MHC independent manner. So there is no need of MHC, major histocompatibility complex. That is not needed. And here comes the advantage. So what is CAR T cell therapy? It is officially known as chimeric antigen receptor T cell therapy and is a novel treatment for cancer, not only for cancer, for most of the autoimmune disorder. Here, what we are doing, we are engineering or synthetically, we are expressing one receptor in the surface of the T cells, which is present in the malignant T cells. And what we will target this T cells to bind to the malignant tumor cells. Malignant tumor cells, especially this CAR cell therapy, is commonly used in blood cancers because the CD19 antigen is present in the surface of tumor cells, and the CAR can bind to it very easily and activate the T cells, thereby distract the cells and can affect the tumor or can reduce the tumor, it can kill the cells or apoptosis signal can be, cascades can be activated. So, how it works, how this CAR cell therapies works. CAR cell therapy works, the first step is we are collecting the leukocytes from the patient. Can anyone explain what is leukophoresis? Leukophoresis? Please unmute and say, can you please say leukophoresis? It's nothing but the process of collecting the blood cells from the patients. And what happens is we will separate the cells and then reinfuse the blood back to the patients. So leukocytes, T cells are separated, and what happens is this T cells is taken, and the next step is what we will be doing, modification of the T cells. We need to express the chimeric antigen receptor. So how we will express the chimeric antigen receptor? Any guesses? How we will express? This receptor has to be expressed in the surface of the T cells. Which way can be used? Can you please unmute and anyone answer? We can make it this more discussion type of webinar. So please unmute. We can express with the help of vectors, either viral vectors, especially the vectors that is collected from the vectors that belong to this group is lentivirus, or without virus, we can express this particular chimeric antigen receptor. So virus can be used, and the most novel technique of using this thing is, I mean, expressing the chimeric antigen in the surface of T cells is. Have you heard of Cas9, CRISPR, CRISPR-Cas9? That method can be used. That is a genetic editing tool which can be used to express this particular receptor in the surface of tumor cells. So now first step is leukophoresis. The patient's blood, the T cells are collected. Now the receptor is expressed in the surface of the T cell. Now what do we have to do? We need to get back to the patient, isn't it? We need to give it back to the patients. So for what that purpose, what we will do? Can you say before that what we will do? Once it is expressed, we will culture it in a lab, and after that we will reinfuse it to the patient. So from the collection to the reinfusion, a time of 5 weeks or 6 weeks is necessary. So that is T cell and genetic modification. Then targeting the T cells. The CAR on the modified T cells, they will specifically recognize and bind to the protein that is present in the cancer cells and will detect the cancer cells. Then after genetic modification, we are culturing, multiplying and giving it back to the patients. So the patients has to be given one immunodepleting therapy as a primer just before giving the CAR therapy. Cyclophosphamide is commonly used and it is infused and monitoring will take place. What is this infusion? Infusion, after the infusion, there is a chance for so much drawback systemic toxicity to take place sometimes. For example, cytokine syndrome or we are saying neurotoxicity will take place. So that post infusion has to be monitored. uh, all the things that I explained there, you can see this figure. Just observe this figure, you may be able to understand. T cells are collected, returning back the blood. T cells are separated, genetically altered by using different techniques, either viral or non-viral techniques. Novel technique is Cas9. Then the CAR cells are cultured in a flask, suitably reinfused. Before reinfused, a lymph depleting chemotherapy, especially when cyclophosphamide is given to the patient and the CAR cells are reinfused. Here in the slide, you can see the structure of CAR, chimeric antigen receptor. The structure consists of ectoderm, transmembrane domain, and the endoderm. Ectoderm consists of antigen binding site and it also has a spacer. Transmembrane domain is there and an intracellular domain which has signal transduction is there. That is a simple diagram which explains the mechanism of action of this curve. What happens is once the cut comes in attached to the tumor cells can you can see in the right side and the T cells you can see in the left side. First, the activation of T cells occurs because the third portion of the CAR receptor is signal transduction. So that will activate the T cells and the activated T cells will secrete perforin and granzyme. Both the enzymes are essential for apoptosis. One causes proliferation and the other will causes the destruction of apoptotic cells. So what happens is it will bind to here HER2 antigen. It will bind to that and causes the cell death through various apoptotic mechanism, either caspase dependent or independent. So we need to learn more about the structure of this term. As I told you, the structure mainly consists of an antigen binding domain, which in turn consists of a VL chain, a variable light chain, as well as a heavy chain, which is connected together by a linker, and just below that, a spacer is present. You can see the normal T cell, the mechanism of action is always MHC1 and MHC2. Major histocompatibility complex is activated and the destruction of tumor cells will be taking place. MHC is present in the surface of the tumor cells, that antigen present in the surface of that antigen. So what our T cells, CAR T cells are doing? They are in an MHC independent manner. They are targeting the tumor cells. So this first structure, antigen binding domain, it always gives a specificity to our task. It helps the affinity, the ability to bind towards T cells, and the avidity, that is the stable complex formation, and efficacy, the ability to produce the response to the tumor cells. All are because of this antigen binding domain. And as I said, it consists of VH, VL, which is connected by linker, and it forms a single chain variable fragment. This SCFV is very important because the in the CAR cells, the SCFC will bind to the antigen that is present in the tumor cells. The epitope, antigen will have epitope. You all may know about the epitope. So it will bind with this epitope in the antigen. This SCFV. So this mechanism is MHC independent cell activation and affinity, same target, sometimes it will show affinity. So that will depends upon epitope location always, and also the amount of antigen that is present inside tumor cells. Depends upon that only the CAR action will be produced. Then SCFB aggregation we need to take into account, because excessive aggregation can cause sometimes problems in the assortion of T cells, and it will also cause the expression of CARs also. It will, in the T cells, the CARs has to be expressed now. That also will be affected by this excessive aggregation. So in most of the cases, we are preventing that aggregation by various molecular techniques or genetic tools, and we are using a preventing. That should be, excessive aggregation should be prevented. Then comes our next important structure, that is the spacer, which will which will helps to recognize the epitopes. As I told you, it always helps to recognize the epitopes, and that is the main function of our hinge. Then hingers have been derived from mainly CD8 cells, 28 cells, and also from various types of immunoglobulins, especially G immunoglobulins. But the point which is to be noted that the IgG derived spacers, it will cause inactivation or depletion of CAR cells. So when we are using this IgG derived spacer, we will inactivate or mute the mutations of the IgG will be take place to minimize the action with the Fc gamma receptor. And Hinge also has the ability to purify, quantify the CAR-positive cells. There are so many things are there. Then spacer regions, more efficient quantification and purification of the cells are possible due to this. So many advantages are there. Then comes our second structure. So in the CAR structure, the first one is the first one is the antigen binding site and the spacer, which consists of the first ectoderm or the extracellular domain. Now the transmembrane domain. The function of transmembrane domain is it anchors this extracellular portion to the cell membrane. T cell, it has to be, the CAR has to connect in the T cell. It is expressed and now it has to be connected to the T cell. So this transmembrane domain always acts as an anchorage to connect the CAR domain to the T cell. So that is the peculiarity. And CAR expression is also mediated by this transmembrane domain. Assembly card, activation card, clustering, all are maintained by this transmembrane domains. Also, in certain types of card, we will discuss about that. First generation, second generation, third generation, like that, cards are there. So in one type of card, cytokine is also, release of cytokine is also associated. So this transmembrane domain is responsible for the release of cytokines, and this cytokines will activate the T cells, and this cytokines will selectively kill the tumor cells, or if we are saying about the autoimmune disorders, the auto antibody producing cells will be there now, that will be killed by this cytokines, distracted by this cytokines. Then the cars also derive from the same protein that our spacer or hinges derive, either IgG or CD8, 28, etc. Come to the structure, as I told you, CD3 tau is there, CD3 tau, CD4, CD8 alpha derived, CD28 derived will be there. Then each one has its own specific properties are there. If you're taking CD8 alpha or CD28 derived transmembrane domains, the peculiarity is that they have more membrane stability when compared to CD3 tau derived transmembrane domains. Next is the endodomains. Endodomains, the function is that while the ectodomains, it is responsible for directing the T cell to the target, and the transmembrane anchors the CAR to the T cell membrane. The role of intracellular domain is to transmit signal to the T cell. And The signals are transmitted to the T cell. T cell activates and different mechanism, the destruction of tumor cells will be taking place. For efficient response, one point is needed. Co-stimulatory signals are also needed. So if you are taking the case of vast majority of FDA approved CAR T cells, they have a co-stimulatory domain. This co-stimulatory domain is very much essential. It helps for the T cell differentiation. Then it activates the T cells metabolic cycles. It will activate the apoptosis of the tumor cells and also can cause the cell destruction. So that structure, that internal intracellular signal or intracellular domain structure with co-stimulatory signals are highly essential for destruction of the tumor cells. The apt mechanism is taking place in that cushion. You can see the regulatory determinants of CAR T cell function here. As we said, SCFP domain is the important thing. Besides that, hinge is there, which is another important portion. Transmembrane domain is there. Intracellular domain is there. You can see all the CAR T cells. Then sometimes it is attached with the cytokine releasing domains also, which helps for the expression of the increased expression of CAR T cells as well as destruction of the uh, autoimmune cells or the autoimmune antibodies or the tumor cells. So this is the structure. To the history of CAR is also very interesting actually. It starts in 1989 when the first generation cards has been introduced. Then the second generation on 2002. Then second generation for leukemia in mouse model is used in 2003. Then first successful clinical trial of CAR T therapy is in 2011. 2012 is the third generation. Then first successful clinical trial is in 2013 and it is for acute lymphoblastic leukemia. Then in 2017, Novartis Chimera, FDA approved Chimera. It is for the treatment of pediatric as well as young. Mainly, this CAR is used for relapsed or refractory B-cell lymphoma, the main application. So Chimera is used for that purpose and also ALL. Then coming to 2019, Bristol Myers has been treat RR, that is refractory or relapsed large B cell lymphoma targeted therapy, CAR T cell therapy was introduced. Then 2020, FDA approved the CAR therapy Tecardix for adult patients with mantle cell lymphoma. It is nothing but the rare case of non-Hodgkin's disease. Then 2021, Abicama, that is for multiple myeloma, has been introduced. And in 2022, Carvykit was approved for multiple myeloma. Again, refractory, relapsed multiple myeloma. This is also, all the things are similar as we discussed. In 2023, one mutated C kit is added to this. For what purpose is treating the blood cancers with CAR T cell therapy is easy because in the blood cancer, a specific type of antigen is always expressed, CD19. But coming to solid tumors, it is very difficult to treat that solid tumors because a more number of antigens will be expressed and the environment is so sophisticated and complicated. So in that case, we have muted the CAR with c-kit. C-kit G we have muted. It is used for solid tumors. It has been used, clinical trials has been done already. It is very good to treat solid tumors. The what half-life of the therapy is increased, then destruction of the tumor cells ability has been enhanced also. So, to the types of CAR T cell therapy, there are 5 generations out there. And these are based upon all this helps for the tumor elimination and is associated with various signaling pathways also. The intracellular domain always works as the functional endpoints for this classification. And the mechanism of CART, it is MHC, non-MHC dependent mechanism, mainly. Signal cascade. So coming to first generation, nothing first generation, the peculiarity is that it has an intracellular, it has an extracellular domain, it has a transmembrane domain, and it has an intracellular domain. But the intracellular domain doesn't have a co-stimulatory domain. As we already have said, co-stimulatory domains always help for what, for different T cell activation purpose. It is highly essential. So this first generation doesn't have co-stimulatory domains, and also it doesn't have sufficient interlocking to the region, attaching region also. That is one of the major drawbacks. So we have to provide exogenous IL-2 to for the efficient response. So it has less T cell proliferation and also short in vivo lifespan. That was the main drawback of first generation. Only the difference is in the structure. It doesn't have a co-stimulatory domain. Coming to the second generation, it has adequate proliferation. The peculiarity is that it has extracellular domain, it has transmembrane domain, and also it has the intracellular domain. Along with that, a co-stimulatory domain is also there, which helps to increase the lifespan of the conventional CAR therapy. The new therapy, it is highly efficient. It has co-stimulatory signal, so has better cytotoxic properties. Coming to third generation, the peculiarity is that it doesn't have enhanced efficacy when compared to second generation, but it has multiple co-stimulatory signals and better tumor elimination and long-term persistence is also there, but no enhanced efficacy. Coming to the fourth generation, it is very important. Universal cytokine mediated killing. It is now used for all the blood cancers, autoimmune, all majority we are using this struck CAR cells because of cytokine mediated killing is there. It is commonly used. And also NF80 attached domain will be there, and that mediated is there. And the TRUP CAR-T is the well-known used for majority of the blood cancers. Coming to fourth generation, coming to the fourth generation, also it has enhanced efficacy, then less systemic toxicity is there. And the trucks of fourth generation what happens is a secondary trans gene induction will be there and transcription will be increased. By this approach, the secreted signal, it not only stimulates the CAR T cells, it helps to memorize the T cells also and also reactivates the immune system. So this CAR is the important thing, or it is a very novel approach and very good approach for treating a large amount of disease because it has that cytokine region which enhance its efficacy. The last one is the new one, that is nothing but the fifth generation or the next generation. And the main advantage of this next generation is that off-target and off-tumor toxicity can be reduced. CAR, when we use, so there is a chance for off-target toxicities to occur because it will have the ability to bind with some other targets also. So it will cause problems. It will enhance the inflammatory markers and responses. So that off-target and off-tumor toxicity can be minimized by using this fifth generation. It is nothing but the card. It is attached to IL-2 receptors. Along with that, a signaling pathway, especially we all know about the signaling pathway, JAK-STAT pathway. So that activation is taking place. and in an antigen dependent manner, the uh, cells, the tumor cells or the autoimmune cells are distracted. So many other approach has also been there in this field, such as off switch, on switch, like that will be there. That is, a particular drug will be attached, and it leads to CAR depletion or CAR activation. That is there. Then lenalidomide gated CARs are there. So many things are there which enhance the therapeutic window as well as the safety profile of the CAR. We can see the first generation, which does not have a co-stimulatory domain. Second generation, which has a co-stimulatory domain. Third generation, which has multiple co-stimulatory domain. And the fourth generation, which is the important one, truck one, which has, it is known as angered cytokine actually, because it has cytokine transgene that will helps to enhance its tumor reducing activity, tumor killing activity, tumor destructing activity. So I think the MOA in detail also we need to have an idea. Already we had seen the MOA. Just comes the T cells with the CAR comes in attached to the antigen and destruction. And this is mainly occurring in three mechanisms. The first one is degranulation, which is taking place in antigen positive cells. And the second one is through fast passage mechanism. That is, for some antigen negative cells will also be there. No, destruction of that occurs through this fast passage and the cytokine production. That is an important thing which helps for the elimination of tumors. So these three mechanisms explain the mechanism of action of CAR T cell therapy. Degranulation, as I told you, granules, perforin granzymes will be released and it will cause the destruction of the cancer cells. Spell is nothing but it is a mechanism. It activates, and when specific antigen is there, it will activate and it causes the destruction of antigen negative cells that are present in the tumor surface also. So, antigen negative cells will also get distracted. Then our important mechanism, that is cytokines. It will trigger anti-tumor immune response. It activates certain cells in the stroma and will reprogram the stroma that is present, the tumor stroma that is present. So this sensitization of this trauma is considered as an important step in our CAR T cell therapy. Here you can see this three axis, how the three mechanisms, the important mechanism of action of how the uh, tumor cells, especially we are saying about tumor because cancer, a large amount of application is in cancer. So CAR T cell therapy, the MOA you can see in this figure. Cytokine secretion by perforin granzyme axis, then you can see it. Next is our, which all are the FDA approved CAR T cell therapies so far. So far, the approved CAR T cell therapy is the first one is tisagenlecleucel, which is Chimeran, which is used in B cell leukemia, lymphocytic leukemia, and also for ALF. The next is acicaptagen, then lucid. Then next is brusocaptagen, lisocaptagen, maraleucel. Then idicaptagen, vicleucel. Then siltacaptagen, autoleucin. All the different FDA-approved CAR T therapies that is available, mainly for B cell non-Hodgkin's lymphoma as well as multiple myeloma it is used. As I told you, refractory and relapsed leukemia, I mean lymphoma is always treated with the help of this FDA-approved CAR T cell therapies. And coming to our India's first approved CAR T cell therapy is October 2029, and that is known as NextCAR 19, and it costs 40 lakhs. It is actually somewhat less cost when compared to other therapies because other therapies is costing 4 lakh $20,000. The expense is due to its complex manufacturing process and research process. President of India launched the first homegrown gene therapy for cancer developed by IIT Mumbai Tata Memorial Center and ImmunoAct, and that is NextCar 19. Applications of CAR T cell therapy, it is used in cancer and also it is used in other purpose also. Beyond oncology also it is used. It is used in fibrosis, then it is used in, even we can use in allergy asthma, then even we can use it in COVID-19 also. Then type 1 diabetics, all we can use. Autoimmune disorders, we can use this CAR T cell therapy. This is the so far the studies that have been conducted, clinical trials that is going on for solid tumors. As I told you, it is very difficult to treat the solid tumors, and the conventional CAR is not suitable for treating solid tumors as it has more than one antigens and also it has a complex environment. So we are using mainly fourth generation CAR, that is the cytokine mediated CAR for this. Truck CAR is commonly used for this solid tumors. CEA, that is embryonic antigen CAR therapy, then CD276, then CD2041, BPX CAR T cell therapy, all are those used. These all therapies are used for pancreatic cancer, then for different types of prostate cancers, adenocarcinomas, and also studies are going on. Not FDA approved drugs are there, studies are going on. Then also anti-HCD CAR PBL studies have been going on. Forest CAR cells for breast cancer studies are going on. Thyroid cancer also studies are going on. Thyroid cancer, CAR cells GFRA4 antigen detected molecules associated CAR cell therapy is going on. Then CD133 CAR vector constructed brain tumor for gliomas. That also studies are going on. It is not, FDA doesn't approve the final drug didn't came, the clinical trial is going on. This is just what is happening in the case of solid tumors, I said. What is happening is that one NAP secretions will be there, protein secretion will be there, and killing will be there. Immune cells will be recruited to particular region. Then cytokines will be released. Dendritic cells will be phagocytized. Then dendritic cells migration will be there. The peculiarity of the solid tumor is that it is heterogeneous antigen expression is there. When CART is given, what happens is CART along with cytokine domain is given, it will cause the complete destruction of the tumor cells, that particular tumor cells. Next is hematological malignancy. Also, it is used. The hematological malignancy, the common uses for B cell lymphoma, it is used. Diffuse B cell lymphoma, it is used. CD19 CAR T cell therapy is used. Then IV infusion in subjects is then mainly CD19 is used for all the hematological malignancies. Then BARD regimen is there. That is, you may be knowing about that regimen, cancer regimen, dexamethasone is there along with that clarithromycin, and that regimen is there. BRD regimen is there. Right, that regimen is there. So for that regimen also, along with that regimen, you can use this BCMA CAR. BCMA CAR is another B cell modulating antigen CAR therapy is there. So that therapy can be used all for multiple myeloma. So we can use the CAR cell therapy for acute lymphoblastic or lymphocytic lymphoma, non-Hodgkin's disease, all we can use. Studies have been going on. And the recent advance in this CAR T cell therapy is instead of using the complete T cell, not the T cell is used, only the exosomes that is derived from the T cells are used. So CAR, it is attached to the exosomes that is used, and it is the recent advance that is there. It is testing in human breast tumors actually. It is more safer because cell-related toxicity will not be there. So more efficient and more toxicity-free it is. CAR cell-derived exosomes. That is the recent advance in the field. You can see here CAR T cell exosomes, how they also release all these enzymes and how they causes the blubbing in cancer cells and finally destruction of the cancer cells. So this EVS is the recent thing in CAR T cell therapy. So I know our heading is revolutionizing the cancer therapy. But cancer therapy it has an important role in hematological cancer and also in solid tumors. So many studies have been going on, and about six therapies have been approved by the FDA for not for solid tumors, for blood cancers. Beyond that, autoimmune disorders we can use, and also fibrosis disease we can use this CAR T cell. This is how we can use it for the autoimmune disorder. You can see just what happens is the mechanism is CAR based. CAR is nothing but chimeric antigen receptor, auto antibody receptor it is. So CAR T cells are used for this purpose, and it will act through the MSC and execute the background disturbance are there. Can you mute? It can cause this grand enzyme and foreign to release and mechanism of action will take place. Then you may have heard of cartridge. That is also the no which is used for autoimmune.
रख दिया उसका नाम तो रात को कोई सी ने मना कर रहे हैं आप क्या मन्नत और मन्नत का मरेगी मन मन्नत क्यों हुआ मन्नत करता है मन्नत को भूल कर दे मन्नत मैंने रखे कभी मर जाना पूछ लियो ना माने नाम की माने रहमत रख लो बीबी रहमत सारे मोबाइल में बता अच्छे अच्छे नाम आ रहे हैं और क्या आई रहमत बुरा है मेरा रख लो
Excuse me, please note, can you please note it?
माने देख कर रखा मीरब के मीरब रख लो मीरब यही तो बना रही नाटक मैंने
For autoimmune disorder, another thing is also there cartridge. Cartridge is not
चल जाएंगे सही
CAR T cell is not, I mean, CAR-T is used for autoimmune purpose. That is nothing but chimeric auto antibody receptor T cell therapy. Auto antibodies, it is detecting and distracting. And the CAR drug is also used in which the cytotoxic property of the T cells, it will be converted to regulatory property and auto antibodies are distracted. So these two techniques are commonly used for autoimmune disorders. So we can also treat type 1 diabetes disorders by using this, ulcerative colitis using this. Then even in HIV, CAR therapies are there. CD8 plus T cell mediated, CD4 based approach is there, and also one broadly neutralizing antibody is there. That is DNA based. That is a novel approach. Both approaches are there. Both approaches will kill the infected CD4 cells actually. The CD8 plus T cell mechanism, I mean CD4 based approach we can use, and also DNA based approach is used. As we know that mainly glycoprotein 120 is involved in this, our antigen region of HIV envelope. So that is targeting, and this new generation anti-HIV CARs, it is broadly neutralizing the antibodies, as I said, GP120 as well as GP41 motifs are neutralized. Then even for COVID-19, CAR NK therapy approaches are there. NK cells are activated and it is used to destruct the SARS-CoV-2 as well as CoV-1 infected cells. So even for COVID-19, we are using now the CAR T cell therapy. So these all are the applications of CAR T cell therapy. Even infections for autoimmune purpose, for allergic reactions. This is all beyond cancer. Even for vitiligo, studies have been going on. Then viral infections, HCV, HIV, all studies have been going on. Different clinical trials have been going. This is nothing but modified CARs. Some cases we can modify the cart with suitable monoclonal antibodies, and that is there. Then second one, as I said, for autoimmune disorder, chimeric autoantibody receptor is there. Then receptor based CARs are there. Then I told you the main mechanism of CART is MHC independent, but in some cases we need this MHC dependent. So in that case also CD8 plus 4 plus MHC based CARs are there and CARs are there. These are the different CARs that are available, CAR T cell therapies that are available. What is the future? Future is actually there is a very good future for this CAR T cell therapy because it can boost your immune system with a minimum of ADR. So there are it can be used for all the disorders. For autoimmune disorders, it has promising results. For cancer, anyway, it has shown results. For HIV infections and for all these disorders, it is showing a very important role. So CAR can be used for all for reducing all the impact of all that disorders actually. And what is the limitation of this CAR T cell therapy? Main thing is the toxicity itself, because cytokine release is all good. It can cause destruction to the normal cells. So that the off-target of tumor ADR will be there. Then sometimes this CAR, since it is target specific, it is easily available to the systemic circulation. So it can cause encephalopathies, and that is another problem. Then high relapse rate sometimes will be there. Systemic toxicity is there. We can solve these issues by using, as I told you earlier, we can use monoclonal antibody attached in CAR, receptors attached in CAR. All can be used to modified CARs can be used to prevent all this or to face all this limitation. There's a chance for antigen escape. That is the area. I mean, infiltration. That is the challenge. Then toxicity. Then sometimes immunosuppressive tumor microenvironment will cause the destruction or reduction in the action. That also is a limitation. That also is a main limitation of CAR T cell therapy. Now, multiple strategies are there. That is more than one united together to form CAR T cell therapies are there, which target various antigens. So that is there for oncology as well as for other purpose. This more than one united will help for targeting more than one antigens and also can be suitable for all types of even malignancy and also for all types of majority of disorders. So these are the references. So far we discussed CAR T cell therapy is nothing but it is an immunotherapy in which the T cells are modified in such a way that it is expressed with chimeric antigen receptor, and the expression is done by using vectors or non-vectors, and it is infused back to the patients. And we just discuss about the structure of CAR, which consists of extracellular domain, transmembrane domain, and an intracellular domain. And based on this three, the card has been divided into different generation. First generation without co-stimulatory signal. Second generation with one co-stimulatory signal. Third generation with multiple co-stimulatory signal. And the fourth generation with cytokine releasing domain is also there, which is known as trump TART, that is used for majority of the purpose. And the application of CART is very vast. It is used in cancer because CD19 antigen can easily be detected and chimeric antigen can binding site can bind and cause the distraction and activation of T cells and the distraction. Besides that, it is also used in solid tumors, different types of solid tumors, and also autoimmune disorders, even infections, HIV infections, and all. The studies have been going on with modified CAR T cell therapy, not conventional. Modified means sometimes monoclonal antibody is added, sometimes auto antibody receptor is added, or particular receptors will be added. And addition of all this can cause some changes in the structure of CAR T cell which promotes or which helps it to reduce all these disorders. And it is a promising tool. The main limitation is its cost. Maybe we can after some time so many what as the time goes on we can say the expense will also get reduced because the for the manufacturing process it will become somewhat diluted and this expense also will get reduced. So it has an important role, so much application, a promising for cancer and beyond cancer.